Hi guys, I'm Emma Scott and I'm here to run through the group problems with you today for our group meeting. Our group has previously discussed how to do these problems and we agreed mine with you. I apologize ahead of time. Sometimes I cut out. This is my second recording. I have a cold and I think that's part of the reason, but bear with me. We are going to find not that one. We want the other one. Okay, so we're going to go up to the top. We're going to go from the top down and solve these problems. I'm going to be brief on some of these. I'll explain them. If they're done the same way, I'll just give you the answers for the rest of the parts. So for part one, we're performing a cross. We're giving the parents up here. And we know that they're going to all assort independently, as Mendel has told us. So for part A, if we're looking for a specific genotype, we're just going to do a filtering method to find out what the proportion is for probability of each of the individual combinations. So you go and you make a Punnett square and you say, okay, both parents have AA, and we're listed up here. If you do a Punnett square, you find out that half of them are going to be this heterozygous. So you put that there, and then you're going to want to find out what proportion are likely to be homozygous dominant for B. And if you do that with a Punnett square or in your head, depending on if it's easier, you find out that 1/4 of them are going to be homozygous dominant. So you're going to multiply that by your previous answer. And for CC, recessive, homozygous recessive, you know 1/4 of them are also going to be displaying that genotype. And for D, half of them will be, which is kind of surprising unless you do the Punnett square. You might not think it at first. And E, it's half again. Multiplying all those together, you get 1/128. That is your proportion. The same process is used for part B, C, and D. So you take your parents and then you look at the ratios of the end result and you multiply those together to find that specific combination for all the traits. So you get 1/256 for part B, 1/64 for C, and 1/32 for D. For part two, question two, it's a fill in the blank, so we just have to remember terms. So they want to know what paired homologous chromosomes are called. There are two different names for them, bivalents and tetrads. If you think about it, you have homologous chromosomes and they're linked together. That would be two homologous chromosomes, or two chromosome sets that have already been duplicated, joined together, so that is where bivalent comes from. Or if you think about it in terms of how many chromatids there are, if you separate it, you can separate into 4 different chromatids, and that's where the tetrad comes into play. Now, if you want to know what the structure is that connects a centromere to the microtubules of the meiotic spindle, and you want to know what it's called, you remember that they're saying meiotic spindle's already there. So you know it's not going to be the kinetochore microtubules, it's actually going to be the kinetochore that they're talking about. It acts like a little belt joining those sister chromatids together. For part C, we're given a diploid fruit fly. It has 4 pairs of chromosomes, and so how many chromatids there are during prophase 1 and metaphase 2. During prophase 1, you know that you have 2 sister chromatids for each chromosome. So you take 2 times 4, because we start off with 4 chromosomes, that will give you 8. sister chromatids. And then in metaphase 2, you know it's been divided once, so you should have 4 left over. For D, we're just wanting to know how to find the genotype of a parent without knowing what it was before by doing a cross of some sort. Mendel called this a test cross, and we have to remember it's true breeding and it's known for a certain trait, and that way you can determine the parent. For E, we want to know what a non-functional allele can be called other than the non-functional allele. So if we look in our lesson, it says it's a null allele, which makes sense because a null means doesn't work, it's a dud, so on. Well, a partially functional allele, looking through the options, we believed it was a gain of function allele, as it's changed and it has different function, might not work the same way, but it still functions. So that is what we decided it was. For F, we look at the different laws that Mendel used. If we look in the lesson, the law of independent assortment says that the alleles aren't going to care, I guess, they're not alive, but bear with me, which chromosome they're on. And they're not going to say, oh, I want to always hang out with this little a, and I'm only going to stay with the little a. So we know that if we have different traits, they're all going to mix up in various ways, and so it involves 2 traits. It can be used for a dihybrid cross. Mendel's law of segregation, it involves a single trait. This is when he was doing the monohybrid crosses. So just looking at whether it's tall or short, or if it's green or yellow, it would be segregated because we know it's either going to get one or the other, but it's only a single trait, whether it's tall or not. Part 3, this is the most difficult question in the lesson. I started by looking at the genotype that we start with, and knowing that they assort independently, I decided to go through and try and find all the combinations that we would end up with at the very end with the gametes. So you know that the big T's, there are 2 that are big T's, so you can only have a dominant T. We don't have any littles to work with, so all your options are going to have a big T. Then you go to the R's. You can have either a big R or a little r. So if you have a big R, you know it can be paired with either a big Y or a little y. So that gives you 2 of your first possibilities. And then you know that you still have the little r to worry about. So you say, okay, if there's a little r, it can be paired with the big Y or a little y. And that gives you your remaining possibilities. So if we go back to part A, all we have to do is diagram the different positions these can be in to come up with these different combinations. So we're looking at metaphase 1. So it hasn't divided yet, it's just lined up on the plate, which helps us know how to arrange our chromosomes. Since it's a homologous pair, we'll know that 1 pair comes from our mom, 1 comes from our dad, and the different alleles are found on the different parents. So we can have the big R from mom and the little r from dad, or vice versa. So we're going to assign those to the chromosomes so we can keep track of the different combinations. I drew the diagram, and I'm going to show you what that looks like. 1 second. So the diagram that I made looks like this. You take all the different possibilities. So you have your big T's together, and that's always going to be the same. And you say, okay, the big T can be with a big R and a little y, and that's the top. Or the big T could be with a little r and a big Y. And you keep going through the different combinations till you have all of them that we found in B. If you remember, these are going to split along this line. So the top is going to be one of your options, for the end result, and the bottom also. Even though it's going to split again, it should have the same alleles present. And that's how you figure out question number 3. Okay, we have 2 more questions left. We're going to go and work through those as well. Oh, we're still finishing 3, pardon me. So we want to know how many sister chromatids are found in each cell during metaphase 1. During metaphase 1, it hasn't split up yet, and we know that there are 2 sister chromatids for each chromosome. We have 3 chromosomes, but they're homologous. They're still, so it makes it look like there are 6 chromosomes. So we would take 2 times 6, which makes 12 for sister chromatids. Now we're asked the same question in part D, except we're looking at a different stage in metaphase 2. So we know they have split up, but the chromosomes are still linked together in a way that it should still be 12. I might be wrong on that, but that's what we agreed on. Okay, part 4. The horse has 32 pairs of chromosomes. So in part A, we want to know how many chromosomes are present in each cell at the end of meiosis 1. If we think about it, meiosis 1 divides what you have in half. So if you start with 32 pairs of chromosomes, you're going to end up with 16 after meiosis 1. If we want to know how many tetrads are present during prophase 1, which is part B, we think about how many we started off with. So 32 pairs, and each pair is going to be a tetrad. So it should be 32 tetrads during that phase. Part C, we want to know how many sister chromatids are present during prophase 1 of meiosis. So during prophase 1, you remember all the chromosomes have been replicated. So we have the 32 pairs, and for each pair there's going to be 4 chromatids. So working that out, we get 64 sister chromatids should be present. In D, we want to know how many sister chromatids are in each cell during metaphase 2. It should be half as many as were found in prophase 1, so it should be 32. In part E, we're wondering how many bivalents are present per horse cell during anaphase 2 of meiosis. Now, if we remember our question 2, we'll remember that bivalents are the homologous chromosomes that are paired together. After anaphase 2, all the separation has already happened. There's no grouping like that anymore, so there are only going to be 0. There won't be any at all. For question 5, we're given the parents, and we're trying to determine different genotypes and how frequent they'll be for their offspring. We're also given a key for what the alleles mean. So if we remember in question 1, when we were doing the forked method and combining all the different options, it's the same with this one, but you have one more thing to think about. For question A, they want to know the proportion of offspring that are going to be tall, round, green, and terminal. So we're talking about phenotypes here. So we have to remember that both the homozygous dominant for the T trait and the homozygous, dominant, it's going to be expressed in those combinations. So not only do we have the 1/4 of the offspring who are going to be homozygous dominant for T, half of them are going to be heterozygous. So we have to remember to include that when we're multiplying, and we get 1 out of 256 are going to be the phenotype that they're looking for. Question B is the genotype again. So it's just like the problems we did in question 1. You multiply their frequencies, and you get 1/32. Same for C. You just look at how often each trait is going to be heterozygous. Out of 16 crosses should be heterozygous for all 4 genes. For question D, part D, it's just like part A. We're looking for the phenotypes. We remember that we're going to have to include all of the traits. I hope this was helpful, and feel free to correct us if we're wrong. Thank you. Bye.